So good morning, everybody. Welcome to the first Oceans and Fisheries Seminar of 2021. I'm Murdoch McAllister, Associate Prof here in IOF, one of the organizers of the seminar. I should start by reading land acknowledgement. We would like to acknowledge that we are gathered today on the traditional, ancestral, and unceded territory of the Musqueam people. So that's the acknowledgement. And I'd just like to say a word about the seminar series. This series is intended to provide a forum for informing people both inside and outside of academia about current research on quantitative fisheries data analysis, and in particular, analysis that pertains there has policy relevance. And I'll introduce our speaker. This is Dr. Ben Nelson, and Ben began his PhD studies at UBC in about 2011. Professor Andrew Treitz and I were his co-supervisors. Ben was supported during his studies by an NSERC grant to the Canadian Fisheries Research Network. That was a 5-year network kind of initiated by fishing industry across Canada, involved collaboration of academics, industry people, fishing industry people on all coasts, even Great Lakes, and government researchers. And Ben was also supported by the grants, for example, Pacific Salmon Foundation during his PhD studies. So Ben, in my view, is a really exemplary PhD student. He is incredibly hardworking, did an impressive amount of field work over a number of years, and also a lot of quantitative modeling and analysis. And Ben's co-authored, I think, at least 3 journal articles out of his studies, and submitted yet other papers for publication. And just this last December, Ben successfully defended his PhD thesis. And Ben's currently a postdoctoral researcher at the Northwest Fisheries Science Center, and an organization, and also at an organization called Long Live the Kings in Seattle. So with that, I'll put it over to Ben.
Great, thanks for the kind introduction, Murdoch. So my name is Ben Nelson. For those of you I don't know, my current affiliation is as a postdoctoral researcher at NOAA's at NOAA's Northwest Fisheries Science Center here in Seattle, where I'm working on some assessment and management modeling for Coho salmon stocks along the west coast of North America. I won't be talking about those models and studies today. I'll actually be taking this opportunity to talk about my recently completed doctoral research, which focused on predator-prey interactions between harbor seals and Pacific salmon in the Salish Sea. So as a lot of you know, Chinook and Coho salmon have supported indigenous, commercial, and recreational fisheries along the west coast of North America for a while. And they're also key prey organisms for other important species like southern resident killer whales. This is especially relevant to the Salish Sea region, where both species of salmon have seen their productivity of their wild populations decrease, and the survival of the hatchery populations drop since around the 1970s. So these 2 panels show the marine survival of Coho on top there, and then Chinook salmon on the bottom. And as you can see, marine survival of Coho salmon dropped from between 10–15% in the 1970s, 1980s, down to less than 5% in recent years. Chinook dropped from 3 or 4% marine survival down to, in most years, less than 1% in the Salish Sea. And a drop from 3 or 4% down to 1% might not sound that profound on the surface, but when you're dealing with the survival of tens of millions of little fish, that change of 1 or 2% in survival materializes in a difference of hundreds of thousands of adult fish, which is critical for supporting fisheries year to year, and then also supporting the prey base for southern resident killer whales. So this is a plot of Coho marine survival along the bottom panel there. And I'm going to show you on the top panel a plot of the harvest rate on these stocks in the Salish Sea. And what I want you to take away is the drastic reduction in fishing pressure around 1995, which is denoted by the blue vertical bar there. And so you go from about 60 to 80% harvest rates down to less than 5% really abruptly in 1995. And then if you see how, how the marine survival in the bottom panel changes, there's really not a marked increase in marine survival as a result of these fisheries closures. And that would suggest that this is probably not a case of recruitment overfishing. And if you look at similar data for Chinook populations in the Salish Sea, you'll see kind of the same dynamic. You'll see abrupt reductions in harvest rates and no really appreciable gain in recruitment or marine survival. So over the same time span, pinniped populations in the Northeast Pacific have increased at impressive rates due to federal protection in the 1970s from hunting and culling programs, particularly the harbor seals, which are shown there on that bottom photo. And in the Salish Sea, for example, in the Strait of Georgia, harbor seals have increased from around 5,000 animals in 1970 to around 40,000, give or take, in as recently as 2008. To the south in the Puget Sound area, there's also been pretty rapid increases in harbor seals, less than 1,000 animals in 1970, and probably around 15 to 19,000 animals, which is suggested by recent surveys. And these animals are thought to be at carrying capacity, which was reached in the 1990s after the populations have leveled off. So you saw a really drastic increase in the 70s, 80s, and then kind of a leveling off at K around the 1990s, and they've pretty much held steady since then. Let me go back here real quick. So the coincident increases in populations of pinnipeds, and then the coincident decline in survival of Chinook and Coho have led some to speculate that seal predation might be the driving force behind these changes in salmon abundance since the 1970s. But it's important to keep in mind that increases in seal abundances haven't been the only major change in the Salish Sea ecosystem since the 70s. There's been large-scale production of both Chinook and Coho hatchery fish that commenced in the 50s and 60s, peaked in the early 90s, and additionally, these hatchery fish have also become larger, particularly with Chinook, and the release timing of Chinook and Coho has also been really narrowed down to like a 2 or 3 week window in May, and so you have really a huge subsidy of hatchery fish that are entering the system at once every spring in both the Puget Sound and Strait of Georgia, and that raises questions about how hatchery populations might affect wild stocks through competition for food and habitat. So for this talk, I'm going to cover 3 primary research questions that I addressed during my dissertation. The first was to ask to what extent do changes in seal populations explain the changes and the variation in wild Chinook populations in the Salish Sea since the 1970s. And I did this while also adjusting for alternative hypotheses like increasing hatchery populations, which also might explain changes in wild productivity. The second question I sought to answer was that if seal abundance and salmon productivity are correlated, I wanted to estimate how many juvenile salmon are being eaten every year, as survival during these early marine months is thought to be, really the bottleneck for overall adult abundance in both Chinook and Coho populations. And then finally, if evidence does exist for a causal relationship between seal predation and declines in Chinook and Coho in the Salish Sea, I asked the question, well, what could we do about it? Could these seal populations be actively managed in the region? And what would some of those alternative management strategies look like? So the first question, are seals and hatchery production associated with the declines in Chinook salmon. This is the first paper I published for my thesis. In case you're interested in the details, it's in CJFAS. So if you have any questions about the nuances of the modeling approaches that I'll discuss, I encourage you to sort out the details there. So to evaluate the relationship between seal abundance and changes in salmon productivity, we might use like a basic statistical model to see if the abundance of predators correlates with survival and productivity of wild stocks. So to do this, we used a Bayesian hierarchical model and included covariates for seal density and as well as hatchery Chinook abundance. And we also evaluated what's called a CAR model, which is a way of spatially relating the covariates in this model. This is a type of model that's often used in epidemiology. And instead of assuming that predation effects or hatchery effects are exchangeable across the entire ecosystem, this type of spatial modeling approach allows kind of like a regional clustering to happen. So it's conceivable that some stocks in certain regions might be affected differently by hatchery effects or seal effects. And so this is a way of controlling for those potential spatial effects. The second part of this analysis, what we did, we built a statistical model and then used that to basically inform an age-structured population model for wild Chinook salmon. And we used that model to simulate a bunch of different scenarios where seal density varied and hatchery abundance of Chinook varied as well. And when we used that to infer changes in management reference points like maximum sustainable yield and the harvest rate that occurs at maximum sustainable yield. So a spatially explicit model, like I mentioned, might be advantageous in this situation because, like I said, it's conceivable that the different densities of seals in the Strait of Georgia might result in different effects on certain populations of Chinook. So for instance, a Chinook coming out of Howe Sound up here might have a different level of encounters with predators compared to, say, a Fraser fish that migrates south and needs to run this gauntlet through the Gulf Islands and the San Juan Islands. Similarly, this is a figure that depicts the contribution of different regions in the Salish Sea of hatchery releases. And so you can see in the South Puget Sound, there's a far higher level of hatchery production of Chinook compared to areas like the North Puget Sound or Fraser River area. And so, it's possible that hatchery effects on these fish might vary from region to region. So to capture the full spatial extent of the Salish Sea, we collated population data on 20 different wild Chinook salmon populations in British Columbia and Washington, and then evaluated dozens of different candidate models that were fit to this data with the goal of identifying which combination of factors explain the most variance in the full data set. And so as you can see in the slide here, we've got really good spatial coverage of a lot of the major Chinook stocks throughout the Salish Sea, and we even included a few on the Washington coast as kind of a way of potentially comparing effects from the coastal populations to the Salish Sea populations. So to jump right into some results, I'm going to show you, this is the top 48 candidate models that were evaluated. On the y-axis is our model selection criteria. The higher the metric here on the y-axis, the worse the model performs. And so the best models are, have the lowest DIC down here. And you can see that the best models contain seals as a seal density as a covariate. And you can see that the models that don't contain seals, there's a really abrupt change in the amount of variance explained by these models. And so, there really wasn't close. The models that included seal density, regional seal density as a covariate explained, a ton of variation in the changes of wild Chinook salmon production since the 1970s. And the best models also incorporate a hatchery covariate, but as I'll show you here in a second, hatchery effects, while they were included in the best model, they didn't really jump out at us as a potential driving factor. So overall, 14 of the 20 wild Chinook stocks that we evaluated showed a significant negative association with harbor seal density over time. None of the 20 wild populations had a negative association, a significantly negative association with hatchery abundance. And interestingly, there wasn't strong support for spatial effects in either of these covariates, which suggests A, we either didn't have the right data to tease out that signal, or B, that really these seal effects are occurring on an ecosystem scale as opposed to like a sub-basin scale. So I then used the statistical model that I just described to estimate how changes in seal density and hatchery abundance would change management reference points on wild Chinook populations. So here we have harvest rated MSY on the top panel and then MSY on the bottom panel. And then this is over an index of seal densities that we've seen over the past 50 years. And you can see pretty clearly that most stocks have a negative relationship between MSY and seal density, as well as the harvest rate that occurs at MSY. Conversely, with hatchery releases, it's kind of a mixed bag depending on what stock of Chinook that you look at. You have some that are negatively associated with hatchery releases, others that are positive, but none of these are significant. So you get kind of a neutral mean effect on the reference points for wild Chinook for the hatchery covariates. So to conclude for this part of the talk, Chinook productivity and yield are negatively associated with a harbor seal density in most populations. There's support for basin scale effects across the entire Salish Sea, but less so at the local scale. And the negative association that we found with Chinook productivity and seal abundance has been corroborated by a recent study that just came out that also showed that Coho is also really, their survival rates are negatively associated with seal density as well. So it appears that both Chinook and Coho in the Salish Sea seem to be significantly negatively associated with seals since the 1970s. And given these results, we'd encourage future assessment efforts and modeling efforts like forecasting to at least consider biotic factors like predation as opposed to just a suite of environmental factors, which is typically done. So in light of the correlative evidence for a connection between seal abundance and declining productivity and survival of Chinook and Coho, I wanted to interrogate this relationship with a bit more realistic mathematical model to see how many young Chinook and Coho salmon are lost to seal predation every year in the first 6 months of life, which is really thought to be the limiting factor for productivity in a lot of these salmon populations. Until recently, quantifying the number of juvenile salmon in the Chinook diet in the seal diet was really difficult, but recent advances in DNA metabaric coding techniques developed by my former lab mate, Austin Thomas, has improved our ability to do so. And this is done by combining DNA extracted from seal fecal material, AKA scats, along with bones and otoliths that are extracted from those scats. And this allows us to differentiate among salmon species in the seal diet, as well as distinguish between juvenile and adult fish in the diet samples. And so this shows the spatial extent of our diet study, which was conducted between 2012 and 2014 at 4 different sites in the Strait of Georgia. And this resulted over 1,400 scats that I used in this predation model that I'll describe here. So to explain the dynamics of salmon populations in the Strait of Georgia, I built a simple kind of life cycle model for Chinook and Coho that accounted for transitions in life stages during the first year of life or so. And my intent was to represent the emergence of salmon from freshwater habitats and into the early marine stage in the spring and summer, and then throughout the first year of life in the fall and winter until their migration out to the high seas. And so the predation model that I developed here, it captures the seal diet and their annual prey requirements, in addition to the number of seals in the Strait of Georgia and how that population has changed over time. I combined a seal submodel with separate submodels that describe the abundance and growth of Chinook and Coho salmon over the first year. And then I fit that model to observed salmon abundance data that were taken from the Ricker Trawl survey data set for the years between 2004 and 2016. And once that model was fit, it allowed us to generate predictions of the number of young salmon eaten every year, the amount of seal mortality that contributes to the overall level of mortality during that first year. So again, to jump to some results here, for both juvenile Chinook and Coho, the top row shows the instantaneous mortality change as a result of seal predation between the 1970s and 80s up through about 2014, 2015. And as you can, and the shaded regions are the 95% confidence intervals. And as you can see, there's really a pretty drastic change since the 1970s to current years. On the bottom, I'm going to show you the proportion of the annual outmigrating cohort that's eaten by harbor seals. So a recent 5-year mean suggests that about 41% of juvenile Chinook salmon entering the sea every year are eaten by harbor seals. And then for Coho, it's even more drastic. About 1 in every 2 outmigrating Coho, we estimate, is potentially eaten by a harbor seal. Now for the caveats, as with all models, our predation model has several key assumptions. There's obviously going to be bias in the diet data from things like differences and biases associated with the sites that we've sampled. And then there's also uncertainty with the bioenergetic models that we use to describe the prey requirements and the bioenergetics of the harbor seal population. And additionally, there is some uncertainty with the freshwater the survival of these young fish during the freshwater stages and how that varies from year to year. But the general conclusions we can draw are that our models suggest that that seals are capable of eating a lot of salmon in a given year, and mortality from seal predation during the first year appears to be a significant component of first-year survival. And then given the changes that seem to have occurred since the 1970s in these predation rates, it appears that seal predation could be potentially a driver of those drops in survival between the 1970s and current years. But however, there is an uncertainty associated with these models and these estimates. But despite the uncertainty in the different aspects of this model, really the key takeaway here is that small diet proportions of salmon in the harbor seal diet don't necessarily mean a small impact. On average, we saw 1 to 2% of the harbor seal diet consisting of juvenile Chinook and Coho salmon in any given month in the spring and summer. But 1 and 2% results in, it suggests that there's millions of little fish that are being eaten every year. So despite all the assumptions that we made and uncertainty, really the take-home message here from this analysis is really not to conflate small diet percentages with small impacts. It appears it's the opposite. So if managers did accept that there was strong evidence of a causal relationship between seal predation and salmon survival, these managers might want to know if the seal population could be controlled as part of either an adaptive management style experiment or a sustainable hunting program. But prior to embarking on these really controversial management actions, it's probably in the manager's best interest to evaluate with the best available data and evidence how these different management scenarios might play out, how long it might take to reach a certain abundance of seals if you did start removing them, and how much effort would be required to implement such a strategy. And so to do this, I built an age structure population model of the British Columbia harbor seal population that was capable of simulating different management scenarios and could be used to compare expected outcomes of each if managers did embark on a strategy of active population control of the harbor seal population. To do this, I developed a age structure population model using a Bayesian framework. It was a state-based model, so we could, control for the amount of variance that was, or the amount of error that was brought into the model just by the observation component and as well as the modeling component. This model that we developed integrated data on seal abundance as well as age composition data that was available from the early 60s when scientists were able to lethally sample the seal population. And it also uses harvest data from the culls and bounties that were taking place throughout the 19th century and then the first half of the 20th century. And so we used that age structure model then to simulate active population control of the harbor seal population. And we evaluated both lethal measures through hunting and harvesting. And we also evaluated non-lethal measures, which in this case would be fertility control or sterilization, which has showed some promise based on pilot studies of gray seals on the East Coast. And we also evaluated like a combined approach that would incorporate both lethal removals of harbor seals and then sterilization as well. So just a little context here. It's important to realize that this seal population has been reduced in the past for hundreds of years. Like I said, by culling and bounty programs in the 19th and 20th centuries, but also, there's also been some level of harvest by indigenous peoples for thousands of years. We don't really know precisely what that level of harvesting was, but it's safe to assume that this population is capable of being harvested and then recovering based on their response to federal protections in the 1970s. So this shows a population reconstruction by Peter Alesiak that was done using the culling and bounty data here shown in the bottom panel. So it shows that these populations can really be subjected to a high effort. 15,000 animals a year have been removed in some years, and the rapid recovery suggests that it probably could be exploited or controlled in a sustainable manner. So moving on to some results for the age structure model. This plot shows the model fit to the data. So on the y-axis we have the abundance of harbor seals from 1880 to present day. The gray bars show the culling and bounty program removals over 100 years or so, and then the protection in 1970 shows the rapid increase in the population since then, The gray circles show the data that we fit the model to. Those data are aerial survey data from British Columbia. And then this is the model fit to age composition data. So on the y-axis is the proportion of animals of the population, and then on the x-axis is it the ages, and for male and females, and really just the take home here is that the model fit the data quite nicely, which is always ideal. So I'm going to show you now some results of the simulations, and I'm going to show you a panel that shows basically the time to in years to a 50% reduction of the harbor seal population. And I'm going to show you the difference in what it would take to achieve a 50% reduction of the population through a management strategy that was exclusively sterilizations versus one that was achieved through lethal removals. And what I want you to realize is that regardless of the management action, it really takes it's going to take a long time and a lot of effort to achieve for either. So with sterilizations, you're really looking at, even if you were to inoculate 15,000 animals a year, it would take at least 10 years to do that. So it's probably not feasible for budgetary and just pure logistical reasons. I think a management strategy that's just exclusively sterilizing animals is probably not going to cut it. With lethal removals here, shown by the series with circles, if it would really take high effort of removals to get that done in under 5 years. You probably have to remove animals at the same rate that was seen at the historical maximum to get a 50% reduction in 5 to 10 years. And there's also, so this panel shows what a combined management action would look like in terms of how long it could take to reduce the population. So this first panel shows a management action that would remove 7,500 animals a year lethally, and then also apply 3,000 sterilizations. And you could actually combine those two measures to get it done in a reasonable amount of time, say under 10 years. But one issue that you introduce by using non-lethal sterilization is that because of the lag it takes, that once the animal is sterilized, once there's a lag between the time the animal is sterilized and then the effect on the population. And so one thing you risk with applying non-lethal measures is this tendency to overshoot with sterilization. And that can result in variation around your target abundance. And it can also result in a lagged recovery. So if you, say for some reason, you wanted to stop your management action because of unforeseen ecosystem effects or what have you, if you're using sterilization, you really have to wait for that group of sterilized animals to exit the population before you can continue recovering. So that's one potential advantage of lethally removing animals, that if you want to stop your management action, it would essentially begin as soon as you stopped killing animals. So to conclude, non-lethal methods in the form of sterilizations are probably difficult to control. Lethal methods are probably faster and more precise, but both of them are potentially viable methods of controlling the harbor seal population, especially in either exclusively through lethal removals or through like a combination of the two. So to conclude, and just to mention again the three questions that I talked about, wanted to address if seals and salmon populations are statistically associated to some level. If they were, how many juvenile salmon exactly are the harbor seal population, how much are they eating every year? And if there is strong evidence for a causal relationship between seal predation and salmon declines, what can be done about that recovery? So results from the correlative approach and then the predation model suggest that there is evidence for a causal relationship between seal predation and populations of Chinook and Coho in the Salish Sea. However, there are uncertainties in these kinds of approaches. And one that I didn't mention is this assumption of additive versus compensatory mortality. So if we could snap our fingers and reduce the seal population by 50% overnight, the seals might be gone, but would that wouldn't necessarily rule out the potential for some other predator to kind of step in and fill that void. And this has been documented down on the Columbia River, where they've made really impressive efforts to reduce avian predation by culling both terns and cormorants. But what they found was that even if you remove avian predation from the downstream migration of steelhead, it's likely some other predator will compensate for that total mortality somewhere downstream, whether it be further down in the river or whether it be in the marine environment. So removing seals, even if there was a causal relationship, isn't necessarily going to prevent some other unknown ecosystem impact from occurring. Both lethal and non-lethal approaches appear to be viable for controlling the harbor seal population, but important trade-offs exist with each. And finally, it's important to understand that while this research is an important step to understanding the role that predation may play in survival and ecology of young Chinook and Coho salmon, there's still a range of issues related to risk management and economics and politics, frankly, that need to be addressed. This research can't do it all. And so it really is kind of a foundation for potentially other research that's related to this, but it's certainly not the end all be all. And I hope that this work can serve as, at least a start for navigating these complex issues in future work. With that, I'd like to acknowledge the people who funded this research, the Canadian Fisheries Research Network and the Pacific Salmon Foundation, and then a group of individuals, particularly Andrew Trites and Murdoch McAllister and Carl Walters, that were really integral in developing and executing this research that I just described. And with that, I'll be happy to take any questions that you might have. Thanks.
Yeah, thanks very much, Ben, for a great talk. So we have quite a bit of time for some questions. So yeah, speak up. So Mike, go ahead.
Thanks, Ben, and congratulations on wrapping up your PhD work.
Thanks, Mike.
I'm wondering what might be known about how much seals move around from one haulout to another in thinking about possibilities of control at local versus ecosystem-wide level scales. So for example, if there was a desire for control to target specific salmon populations that were in trouble, would local efforts at seal culling pay off, or would other seals just move in to take their place?
That's a really good question. And it's a question that I probably should have emphasized, I probably should have addressed in the talk. But this is one kind of issue that I think people are anxious to discuss. Can you go into these different local areas or different estuaries, and could you just remove problem animals, and would that be able to potentially materialize in some significant increase in survival for that local stock? And I think the best evidence that we have right now suggests that that's not going to be the case. And the reason is twofold. The first one being that the predation models that I developed clearly show that predation on these young fish doesn't necessarily occur at the highest rates right when these fish enter the fresh water, that it's really a prolonged level of predation by harbor seals throughout the entire summer and even into the fall, that lasts and that really it results in really high impacts in the pelagic environment too. So this probably isn't just a case of fish getting eaten right after they are released from the hatchery. It's actually more complicated than that. It's actually likely that a lot of predation is occurring once these young fish have moved offshore and are interspersed with other prey species like herring. And so that said, I think even if you were to remove problem seals or a group of seals from one area, I think it's highly likely that mortality would be compensated for somewhere down the line.
Great, thanks.
Hey, Ben, thanks for the presentation.
Sure.
Just a quick couple of questions. The first one, I was just wondering, what did the Chinook population data consist of?
For which part of the analysis?
So let's say like when you were looking at abundance of Chinooks across different spatial regions. Yeah.
So for that, like I said, we had 20 populations of wild Chinook from the Salish Sea and Washington coast, and basically we derived the productivity for each of those stocks from a combination of spawning surveys and then exploitation rate data that were published in like the PSC reports. So basically the 20 stocks that we used were by and large the indicator stocks that the PSC uses that are pretty intensely monitored for the mixed stock fishery that occurs along the coast.
Okay.
And there are a couple of questions in the chat box.
Regarding the hatcheries, the impact of hatcheries. So could there be other covariates at the subpopulation level that could explain why the impact of hatchery was positive in some cases and negative for others?
Yeah, I think so. A lot of people have been interested in the hatchery component of this work because they look at this problem through the lens of, oh, well, if there's no effect of hatchery fish on wild fish, doesn't that just mean we can pump the system full of hatchery fish for to feed southern resident killer whales? And I really hesitate to use our study as grounds to do that because the covariate that we included in our models was just hatchery abundance, and then the response variable that we measured in the wild Chinook populations was just productivity. So even though our analysis didn't find any significant negative impacts per se on productivity of these fish, that's not to say that there isn't some other impact on wild stocks that is resulting in maybe smaller fish or maybe more vulnerable fish. So to answer your question, are there other covariates that we could have used to describe the hatchery component? Yes, there are. And I guess I might reference you to a recent paper by Catherine Sobocinski, who kind of, I think she developed a similar analysis, and I think she included some different hatchery covariates that weren't just related to overall abundance.
Okay, thanks. One last one, if I may.
Sure.
I think we have hatchery waiting.
Okay, well, so then if you want to move on, let's go.
You can save that question for later. Oliver, we'll come back to you. Okay.
Yes.
So Ken, got your hand up? Got to unmute, Ken. You're still muted, Ken. Here. Try to unmute you. No, you're still muted, Ken.
How's that?
Okay, there you go. Good.
Good. Excellent presentation. Thank you. I've got a couple of questions, which Carl and I have been talking about over the last couple of years, is to get some studies going on adult consumption in the Gulf of Georgia and the Fraser. And my thoughts relate to a recent study at the last summer released by the Washington State Fish and Wildlife, showing 3 to 5 adult Chinook were consumed per sea line per day on the Columbia River. And just doing a simple math with 40,000 seals in our Gulf and just Gulf of Georgia only at 1 a week, that's 160,000 a month times 5 months is pushing 1 million. So somewhere between 0 and a million, there's got to be a pretty large consumption. Your thoughts on that?
Yeah, so unfortunately, we can't do it all, and I think that this research kind of, like I said, it's a foundation, and it leaves a lot of questions yet to be answered. The impact on the adult fish being one of them. Like you said, there's a lot of concern about competition for adult fish, both through from a fisheries standpoint, but there's arguably even more interest in the adult consumption component through the lens of competition with southern resident killer whales. And so I can tell you that the data that we use to estimate the consumption on juvenile salmon in this study can be also used to potentially estimate the impacts on adult fish when they come back. And whether people have plans to do that, I can't say. I think, like you said, like you alluded to, there's been some work certainly on the Columbia River to estimate these impacts, and I know there may be some work in the state of Washington to do something similar. But like I said, the reason for the impetus for my research was to get a handle on how many young fish are being eaten every year, because I think the consensus is among salmon biologists now is that really the limiting factor for adult productivity, for adult production, and the abundance of adult fish that you see every year, it really does occur in that first year of life at the juvenile level. So yeah, I do think there's potentially more work to be done, and yeah, I'm just happy that we can address the first part of that here.
Oh no, I understand that. We've been banging away at this for 3 years now, and we're getting some pretty good traction at the political level finally. So, boy, just thanks for your good work on this. That's all I can say.
Happy to help.
Carl, and then Carl Roy, and then a question from Carlia Pantera.
Yeah, just 2 things. One of them, Ben, you did a great job on your thesis, and I'm sorry I fell asleep for your exam.
That's okay. Forgiven.
Yeah. No, you're really looking at 2 quite distinct data sets here. One of them is the spawning stock recruitment relationship using spawning information and recruitment estimates that make some use of the Coho wire tagging data. But if there is adult consumption going on, that would actually be included in the estimated recruitment. So the stock recruitment models are implicitly including adult consumption as well, because it means fish that didn't reach the catches that are used in the recruitment reconstruction. But the other data is quite distinct, is the direct estimates of survival rate from release or ocean entry to the second ocean summer from Coho wire tagging data. And those estimates agree in general about the basic patterns and about the predicted mortality rate increases are almost exactly what you calculate from your diet data. But let me raise one point here, is that I think the biggest uncertainty about this whole thing is about what's called non-additive mortality, not compensatory mortality, like if the cormorants don't eat them, something else is. There's a much more fundamental issue about whether the juveniles being eaten are ones that were going to die anyway because they're diseased or unhealthy for some reason. And that's called non-additivity. It had nothing to do with compensatory change. And for all the calculations that you and I have done, it's that non-additivity is the huge uncertainty. Or we just see an increased mortality because something else like disease or high temperatures is making those little guys available, and the seals are consumption of them is that just a symptom of the underlying problem?
Yeah, I agree completely. I mean, I think that's really potentially the scariest scenario of them all, is that these fish are doomed from the get-go, and you know, seals maybe just are the most efficient executioner out there.
Well, you know, I think key thing here is the only way to resolve that is to remove the predation effect and see if the survival improves. There's no other scientific way to do it.
Yeah.
Roy? Roy, did you have a question?
Yeah. Could you hear me? Yeah.
Yep.
Yeah, I'm just making a point here. Like, this study stuff's been, you know, the seal and sea line problem's been studied to death, and it's just absolutely a crime what's going on. And through the studies that Peter Oleziak did in 1990 up to 1990, you know, was really challenging to read that and find out nothing was done about it. Again, in 1998, he did the study on the Puntledge River, where they determined that 36 seals will eat 3.2 million fry. You know, this was going on all over the coast. It should have been looked at that way, because all they did was concentrate on the Salish Sea for their studies. So, you know, Haida Gwaii should have been, I'm just writing, Haida Gwaii should have been canary in the mine, because we got small rivers and stuff like that, and we were wiped right out. And I was a commercial fisherman up until 1998, and then I became a food fisherman. We're wondering if we should start taking salmon for food. That's how bad it's got. And thanks to the sports fishing industry and their vision to collect money from their users and start hatcheries and stuff like that, we got spring salmon, hooray. But the lesser species are all wiped out, and the populations of seals and sea lions on Haida Gwaii are way over the top. Something's got to be done now. And I keep hearing the term, and we heard it in Bellingham when we're at the Transboundary Conference, you know, let's sterilize them, you know, but that doesn't stop them from eating, you know, and it doesn't sterilize all the animals. So those things have to be really looked at, because the science has been there for well over 30 years now. And I started this in 1981 when we seen a real serious problem in South Moresby. I've been pounding at these doors for a long time. I've been to Ottawa multiple times. I've been to Vancouver. I've been to Newfoundland, wherever there's conferences. And today we're sitting here with a full market ready to go. And yet the politics is still running from the animal rights people, who have a responsibility to start protecting, to quit protecting one species over another. And that's the way the government's got to look at it, as well as you scientists. And I thank Dr. Carl Walters for his continuing support on this, because that's what we need. We need people to understand that we're in trouble. And let's talk about the health of fish and the health of sea lions. We can make it doable, because I'll tell you one thing, after mother's breast milk, seal oil is the healthiest thing you can put in your body. And it's absolutely amazing. But you know what? Nobody's promoting that. All that, you know, we're seeing the Bridget Bardot's run a fashion show in what the hell, in Paris with, you know, blood running off on the runway and shit like that, you know. Those guys got a responsibility to ensure the future salmon, because it's not only affecting me today, it's affecting all my descendants. So, you know, we got to do something now. Thank you.
You are. And Carolina, you had a question about transients?
Yeah, hi Ben. Thanks for the presentation. I was just wondering if
Sure.
you've seen evidence, or do you think that recent increases in transient killer whales in the Salish Sea could play a significant role in controlling the seal populations?
Thanks for the question. I do think it's an interesting question, and I actually did my PhD in the marine mammal research unit at UBC, and I know that's a question that my supervisor Andrew Trites has really been interested in, and I can tell you that that there is the conventional hypothesis that like the conventional wisdom that would say that the recent increase in utilization of the Salish Sea by transient killer whales might be the reason why that the seal population leveled off at carrying capacity. So it does make sense. But I can also tell you that there are reasons for that seal population to reach carrying capacity because of bottom-up effects, because of things like prey limitation and potentially habitat limitation. And I guess one thing that jumped out to me was that you have seal populations along the entire coast that reach carrying capacity around the same time. So from the Strait of Georgia all the way down to southern Oregon, if you look at the seal population trends, they all look the same. They all come up in the 70s and 80s, and they all hit that carrying capacity in the 90s. And while the Salish Sea has seen a lot of transient killer whales, I'm not sure that places like the Oregon coast have seen such frequent use of the transient killer whales as well. And so to answer your question, I don't know, but I think there's reason to believe that they could be playing a role. And then I think there's also evidence to suggest that it might be some other effect that's unrelated to transient killer whale predation that's controlling the seal population. So that's a good question, and I think there's one paper that's been published recently that tries to get at that hypothesis, and I think there are definitely a few other people thinking about that as well.
That includes, Josh, you had a comment about transients?
Yeah, so can you hear me okay?
Yep.
So just with the Oregon question and with transient killer whales, I know that's a huge thing that a lot of us are thinking about, and that the fact that if you look up and down the coast, there's several different groups. If you look at Oregon, for instance, if that question was regarding Oregon, there are different populations of transients that rely on different areas that are, especially between California and British Columbia, that feed on different stocks of marine mammals. So the transients we're seeing in the Salish Sea, there's been 100 new calves in the last 10 years. Is the consensus, it seems to be that is contributed from the predation on harbor seals, that increase in that population. But then if you go down the coast where these harbor seals start to, you don't have the population numbers as we see in the Salish Sea, the transient stocks further south don't really seem to focus too much on harbor seals. And their numbers are much lower. We're just finishing up right now a population minimum estimate for the transient killer whale population south of Oregon and California. And the numbers aren't the same as they are in the Salish Sea or off of southern Vancouver Island and Washington State. So there's likely some, I've seen the mark recapture studies that have come out from DFO that have shown that the population has increased. And one of the findings was that there was a correlation between harbor seal numbers and transient killer whale numbers. So I think taking models into effect, we really need to start to look at what's above the food chain of harbor seals. So you're comparing how harbor seals may affect salmon and how that may how that may affect fishing. But we got to remember that there is a predator above the harbor seal that also, that relies on harbor seals as a food source.
Thank you. And Jakob and then Greg.
Oh. Hey there. Thanks for the presentation. I just had a quick question about how you think this research may be applied to different life history types of Chinook, like the yearling Chinook that may not spend as much time in the Salish Sea, or the sub-yearling Chinook that may arrive later in the season and that have different population trends. I think most of the preliminary stuff you showed at the beginning was based on fall populations, fall run. And so I was just curious what your take on that would be.
Yeah, so you're right. The 20 populations of wild Chinook that we analyzed were all fall run, and we did that, you know, because there, as you probably know, there's a lot more population data on these fall run fish, and a lot less on the spring run fish. And so that being said, you know, this idea of differences between different Chinook populations, I think, is a good one to keep in mind, because you know, I think, you know, these models are great for getting us in the ballpark in terms of how many fish might be eaten in a given year. But I also think there's maybe a more important question of, you know, which stocks are actually being consumed at the highest rates. I mean, are these seals, are they just out there making a living off of eating hatchery fish and ignoring the threatened protected wild stocks like the spring Chinook, or are they actually, you know, are they actually having a significant impact on some of these really valuable stocks like the Fraser spring Chinook that are important for all kinds of reasons? And so I think, you know, it might be wise not to get caught up in the numbers of salmon that are being eaten so much, as opposed to like who is actually being eaten. Is it wild fish versus hatchery fish? Is it abundant fish versus you know, fish that are verging on extinction? I think that's an area of research that might be interesting for the future.
Great points. Thank you.
Greg?
Thanks a lot, Ben. That's for the great presentation.
Thanks, Greg.
I just wanted to ask quickly about data access and whether that kind of resonates with you. I mean, challenges with data access, and whether you could just comment on those challenges if you've encountered them.
Well, anytime you're dealing with salmon data, there's always going to be issues with data access. One, because you know, salmon are, you know, whether we like it or not, just a very controversial species with a lot of political implications involved with you know, the science that is generated. And then, you know, there's also the issue of you know, these data sets are constantly being updated. You know, the data that we use for Chinook, for instance, you know, there's always going to be like a 6-year lag between the current day and then, you know, 6 years ago when the you know, when the fish went out to the ocean first, because those fish have to go out for 3 to 6 years and then come all the way back, and then the tags need to be enumerated, and then the math needs to be done to calculate the different rates. And so those data are always being updated, and the people that curate those data and produce them are rightfully protective of them and want to make sure that analysts like myself understand them and don't misuse them. So to your question, yes, data access can be an issue for this type of analysis, and we did experience that. But, you know, thankfully we were able to, you know, work with enough generous people to really make some headway with the project.
Oliver?
Oh, yes. Yeah, so I think the first question was tied into something that Ken was commenting on, and that was just, you know, do the conservation agencies, which kind of would have to place the success of one species over another, do they like even have the tools in place to consider such interspecies trade-offs? And then just back to the follow-up question from Josh regarding the different life histories. So are you, by run, are you referring to the adult spawning run? And then I'm just wondering if that is the case, then did you include different juvenile sub-life histories in your life cycle model?
So let's see, I'll answer your last question first. Basically what we assumed for our predation model was an aggregate of the population. So we didn't explicitly have sub models for Chinook or for spring versus fall. But at least, you know, I think in this day and age, the contingent or the group of spring fish that come out every year, particularly in the Puget Sound, are just so small that they're probably not going to impact any of our calculations significantly. You know, the part that we might be missing by not explicitly incorporating different life histories is maybe a small size difference in the average size of the fish that's eaten by a seal. But I don't think that if we were to include those, I don't think it would affect the overall conclusions of what we did here. But it's a good idea. I mean, it could certainly be done if one had a particular interest in impacts to like spring fish or something like that.
Murdoch?
Yeah. Oh, Oliver, you had a second question, and then I think we'll tie it up here after this. So.
Yeah, it was just tying back to the conservation agencies. Like, let's say.
Oh, right.
Yeah.
So it was. I guess for, you know, every conservation agency seems to have their own specific interest, and a lot of times those interests are competing, and so I guess, yeah, I think you'd probably have to ask them as far as what capacity they would have to address these questions. I will say that I think that there's, you know, the best attempts to address these problems and these questions are going to be collaborative. I don't think people can be siloed off in their own corners working on, you know, small pieces of this larger puzzle. I do think, you know, like someone alluded to, there's a lot of issues with data access and stuff like that. And so I think it would really be in everyone's best interest if we approach this collaboratively and try to pool resources when possible. I know that my projects benefited from that, but could probably have been expanded upon if we did have cooperation in other places. And so yeah, I think it's something to think about.
Well, thanks very much, Ben, for a great presentation and seminar.